Hello, I'm Sivarajan, and I will walk you through our poster today. EpiRegio is a web server which hosts regulatory elements and its associated genes. What are regulatory elements, or for short, REMs? REMs are regions of non-coding DNA which exercise control over gene expression. Decades of research has shown us the importance of REMs in gene regulation. However, identification of REMs and associating them with their target gene is still unsolved. One of the common approaches to associate a known REM with its target gene is to simply link to its nearest gene, as shown here. While it can often be correct, increasing evidence suggests that this is necessarily not true for all cases. Often, REMs exercise control over genes which are far away, like shown in this figure. Our web server, EpiRegio, provides comprehensive access to REMs and their target genes, which are identified using an algorithm called STITCH-IT. STITCH-IT is an association-based method which identifies REMs and their target genes simultaneously. It utilizes paired DNase and RNA-seq data while simply aiming to identify regions of DNA signal variation which can be associated with gene expression. What does that mean? In the example shown here, the blue density curves represent the DNA signal distribution along the genome. We have three samples, sample 1 with low gene expression and others with high gene expression. STITCH-IT scans and identifies regions of DNA signal such as the one highlighted. In sample 1, we see an absence of DNA signal in the highlighted region and the gene expression is low. In other samples, this region has a high DNA signal and high gene expression. This variation in DNA signal could explain why the gene is silent in sample 1. Hence, STITCH-IT will predict this region as a REM and associate the gene as a potential target. You can find more information about this algorithm in the reference shown at the bottom. EpiRegio facilitates your search for REM gene association through three different search queries. First is the gene query. If you have a list of genes, you can simply use the gene query to identify which are all the REMs associated with this particular gene. Second, if you have a region of interest which you would like to investigate, you can use the region query to identify the REMs located in this region and what are all the genes these REMs regulate. And third, if you already know the regulatory element which you are looking for, then you can simply use the REM query to identify the gene which is regulated by this REM. Irrespective of the query you use, each query returns an interactive table with various information on the REMs and its target gene. We also predict the function of a REM, that is, whether it is activating or repressing the gene expression. We also provide some cell type specific information from the 46 different human tissues or cell types we have in the database. We are already working towards adding more diverse tissues and cell types. If you would like to have more information, please check out our poster or our publication. Our web server is available at epiregio.de. Do give it a try and leave us some feedback on what we can improve. Thank you. Have a nice day.